regulation of endothelial nitric oxide synthase. And this is really important because it drives, the endothelial cell dysfunction, which in the lexicon of modern cardiovascular medicine is the term typically referred to abnormalities in the production or bioavailability of endothelial-derived nitric oxide and the resultant adverse changes in vascular reactivity. And this all kind of stems back to this eNOS, or endothelial nitric oxide synthase. So free radical nitric oxide, which is going to be required, is generated exclusively by enzymes associated with nitric oxide synthase. And we've got different types. So we've got neuronal nitric oxide, endothelial nitric oxide, and inducible nitric oxide synthases. We're going to focus primarily on endothelial nitric oxide synthase, or eNOS, as it is, as mentioned up here in point 1, a key component of endothelial-derived nitric oxide synthase and a key component of cardiovascular associated dysfunction. So substrates for NOS-mediated nitric oxide production are the amino acid L-arginine, molecular oxygen, and NADPH. And in some instances, we see that the cofactor required for nitric oxide generation includes this tetrahydrobiopterin, BH4. So here's our normal endothelial cell, and we've got eNOS, that's our enzyme. We have L-arginine, oxygen, and NADPH with the BH4, all giving us nitric oxide. And then that nitric oxide can go and have its biological effect. In a lot of cases, it's not only a vasodilator, but it's going to interact with reactive oxygen species, and so it's a very powerful signaling molecule. Under certain conditions, nitric oxide synthase may generate superoxide instead of nitric oxide. And this is a process that we see in cardiovascular medicine called nitric oxide uncoupling. And we see this in conditions like diabetes, and it's just where we get this increased redox imbalance due to changes in NADH to NADPH ratio, decreased availability of BH4. All of these things may lead to an uncoupling of nitric oxide. So redox imbalance, decreased BH4 lead to an uncoupling of nitric oxide production. So here's our coupled. We have L-arginine, NADPH, BH4, and eNOS that gives us nitric oxide. But in this uncoupled state, we get this transfer of electrons to O2 to form superoxide, which, as we know from previous lecture, is going to have reactive oxygen-related dysfunction. So we get conditions where we have low L-arginine, a high NADPH, low BH4. We still have our enzyme, but in this case, we get very little nitric oxide, and we get more superoxide that gives us hydrogen peroxide and the other reactive oxygen species, things like peroxynitrate. So that's when you look at the literature and you talk about normal coupled endothelial nitric oxide production and uncoupled eNOS, this is what's happening with eNOS, is that we get this increased oxidative stress and low nitric oxide bioavailability. So what's regulating eNOS? That's what we're really going to focus on, is the factors that are regulating eNOS. And if we look at some of the control mechanisms, there's marked discrepancy among the amounts of eNOS messenger RNA, so the messenger RNA that's going to code for endothelial nitric oxide synthase, the eNOS protein, and then the activity of those proteins. So a lot of discrepancy, so a lot of sites for regulation. So complex regulatory mechanisms at both the post-transcriptional and post-translational level. And we can look at this image here. So here's our nucleus, here's our DNA that would code for the messenger RNA that's going to give us the endothelial nitric oxide synthase. That's got to get exported. We then have that messenger RNA where we then have to make the protein, and that can actually work its way to where we get different activities. So the messenger RNA can be degraded, it can be translated, and we can get the actual protein. If we look here at the beginning, so here's transcriptional control, we can see several things. So shear stress. So endothelial cells within the cardiovascular system are exposed to laminar shear, hydrogen peroxide, HDL, TNF-alpha. Those TNF-alpha is inflammatory. Hypoxia, all are going to regulate this. Conversion of DNA to messenger RNA. RNA processing. So we talked about post-transcriptional control. Things like laminar shear is going to come into play here. We've got transport and localization. May have some impact with things like statins. Hydrogen peroxide also going to be there. Degradation of this messenger RNA. If we destroy this messenger RNA, we don't get our actual protein that can then have that enzymatic properties. So we can have things like inflammatory conditions with like TNF-alpha, hypoxia. Some statins will have different sites of regulation on this here. The things that you really need to know I've highlighted down here. So eNOS expression, upregulation, is regulated by a variety of stimuli. Things like shear stress, estrogen, and insulin are all key things that I want you to know that can increase and upregulate eNOS expression. eNOS expression is then downregulated by a variety of factors. So things like tumor necrosis factor alpha, so TNF-alpha. Other things like high concentrations of oxidized LDL. So we don't want to have high LDL, and there's a variety of reasons, but that's also going to be a negative downregulator, or it's going to have a downregulating effect on eNOS expression. And it's also important to remember that messenger RNA levels, are this balance between gene transcription and the messenger RNA degradation. The messenger RNA doesn't just stick around forever. It does get degraded. So it's, again, this balance between the production, which is through gene transcription, and the degradation. Things like TNF-alpha destabilize eNOS messenger RNA, so it's going to help it become degraded. Other stimuli that have been reported to decrease eNOS include lipopolysaccharides, hypoxia, oxidized LDL. Things like vascular endothelial growth factor, also known as VEGF, enhance the stability of eNOS messenger RNA. And we can look at this image down here, and it really sums up, some of the things that are driving this eNOS protein that we really need to actually get the activity. So we have our gene, we've got things like shear stress and estrogen. They're going to have a positive impact on transcription. Things that are going to have they're going to have a positive impact on degradation, meaning it's going to be more likely to degrade. There's our TNF-alpha and lipopolysaccharides. Vascular endothelial growth factor is going to have a negative effect here in that it's going to downregulate mRNA degradation. So once we've got that protein, we've got some post-translational modifications of this eNOS protein, and this is where a lot of the control comes into play that we really deal with in physiology and in medicine. So in a quiescent cell, eNOS is specifically targeted to small invaginations of the plasma membrane called calveolae. Remember, we talked about these earlier in the semester when dealing with the plasma membranes. Here's an exact example of where this comes into play. So remember, these calveolae, they contain diverse receptors, signaling transduction pathways. So it puts everything in a nice tight area so that it kind of helps speed and facilitate the signaling. So it serves to position eNOS to receive signals from upstream signaling pathways and facilitate communication with downstream activators. So within our endothelial cell and within these calveolae, eNOS is bound to calveolin 1, which inhibits eNOS activity. Disease states in which calveolins have been implicated include things like atherosclerosis, hypertension, diabetes, which, if you look up, those are some of the top diseases we deal with in modern medicine. So here's our plasma membrane, here's our calveolae, here's our calveolin, and here's our eNOS molecule. And when they're bound like this, that inhibits eNOS activity. So this is kind of in a downregulated, downinhibited state. So what do we need to do to get this nitric oxide synthase in a state where we can actually use it? Well, the first is the intracellular calcium PI3 kinase pathway. And then we've got a diverse group of agonists. These include things like bradykinin, acetylcholine, that activate G protein dependent signaling pathways. So we'll kind of go back and forth between our text here and these images. Here's our G protein linked receptor that we've talked about in past lectures. We've got things like acetylcholine that can bind to this receptor, and they activate a variety of steps. So here's our PI3 that activates our endoplasmic reticulum, and that activates calmodulin, that activates nitric oxide synthase. So we can see that in point 11 here. This acetylcholine results in the activation of calcium channels at the calveolae, further activate calcium channels in the endoplasmic reticulum by a phospholipase C and PI3. So here's our PI3, here's our endoplasmic reticulum. This intracellular calcium level is critical because as this goes up, largely due to this increase in calcium through the endoplasmic reticulum, we get calmodulin binding to endothelial nitric oxide synthase, and this disrupts the inhibitory calveolin eNOS interaction. So it gets that breaking up and allows nitric oxide synthase to convert L-arginine to nitric oxide. We'll look at this below here. I like this below image a little bit better. So we have acetylcholine. We get activation of phospholipase C, PI3. That results in an increase in calcium. It binds with calmodulin, gives us our calcium calmodulin complex. Here in this inactive state, so here's calveolin 1. In our eNOS, it's inactive because it's bound up in here, and here is our calveolae. This calcium calmodulin complex binds with eNOS, so it allows this to separate, and once it's in this state, it can convert L-arginine to nitric oxide. So this is how we get from acetylcholine to nitric oxide, and each of these steps is important. This is the signaling pathway from acetylcholine to nitric oxide. There's other factors that can increase eNOS activity. So there's a diverse type of agonist, things like proteins, so vascular endothelial growth factor, insulin, small molecules like estrogen, and mechanical forces like shear stress, that would be stress with the blood running past the endothelial cells. These can all affect eNOS activity through the PI3 kinase AKT pathway. So shear stress is probably one of the biggest ones studied. It's the blood as it travels faster across an endothelial cell. That increases the shear stress, and the endothelium can actually sense that and results in an increase in nitric oxide that causes then shear stress mediated vasodilation. So this shear stress signaling via potassium channels at the cytoskeleton. So here's our endothelial cell. We would have potassium channels in the cytoskeleton which sense this shear stress. We get some tyrosine phosphorylation, and more importantly for this class, we get activation of the PI3 kinase, subsequent activation of AKT, which then increases the affinity of eNOS for calmodulin. So it really promotes this activation of eNOS. So shear stress, PI3 kinase, or AKT, which again in some literature it's protein kinase B, gives eNOS. And it does so because it increases the affinity of eNOS for calmodulin. We saw that. Here's our calmodulin, calcium calmodulin complex. The AKT helps promote this step, which again gives us our nitric oxide. And lastly, heat shock protein does come into play here, and this is really emerging work, so we're only going to just kind of touch the surface, but we know things like heat shock protein 90. It's a molecular chaperone. We talked a little bit about these early in the semester. They're involved in protein trafficking and folding, and they're going to have a key role in agonist-dependent eNOS activation. So I'll just highlight a few things here and then come down to the figure. Heat shock protein 90 binding stimulates eNOS activity by cooperatively enhancing the affinity of eNOS for binding to calmodulin. So it's going to help this calmodulin eNOS binding, which is going to be required to separate it from calbolin 1. Heat shock protein 90 can bind to both active and inactive AKT and is required for the interaction of AKT with eNOS. It increases the rate of AKT-dependent phosphorylation. So it helps increase the rate at which we can activate AKT. And it's important that this comes into play when we start talking about some drugs. Things like statins are used to induce eNOS phosphorylation of AKT. So the statin AKT eNOS interaction becomes really important when you're trying to improve vascular health. So if we look at our image here, so here's our plasma membrane, here's our calvolus. We have things like bradykinin and acetylcholine. We have shear stress as our stimulus. We'll start down here at our Golgi apparatus. So here's our eNOS molecule. Here's calbolin 1. We get this translocated to the plasma membrane. Here's our calvolus. I'm going to come over here to this right. So here's our eNOS molecule. Here's our calbolin. They're bound and it's in this inactive form. We get things like AKT. Here's our calmodulin. Here's our calcium. There's calcium coming from the endoplasmic reticulum over here. They interact. So here's our calcium calmodulin complex. It interacts with eNOS. AKT helps facilitate that. We then work its way over here, and heat shock protein again is going to help with this binding, and we get this nice complex where eNOS is activated and gives us large amounts of nitric oxide. So this is in our coupled state here. So we can see that all of these steps, these signaling, where we've got the calcium calmodulin, the AKT, the calbolin, heat shock protein, this interaction drives this increase in nitric oxide, which then is a signaling molecule in its own right, which will work its way to smooth muscle cells and cause vasodilation.